Welcome back to this next video, and in this video we are going to talk about the ligands and the receptors. These ligands and the receptors, they are the basis of cellular signaling, and all of the signaling mechanism between the cell and within the cell, they are dependent on the ligands and the receptors. When you talk about the communication in the living world, there are two kinds of communication. One kind of the communication is called the intercellular signaling, and this intercellular signaling means that there is communication between the cells, because this term inter mean between. So this this intercellular signaling mean that the different cells, they are communicating with each other. Another kind of the communication in the living world is the intracellular signaling, and this term intra mean inside. That means you're talking about the communication within the cell. So the communication can be intercellular, that means communication between the cells, or that can be intracellular, that means communication within the cell. If you define these ligands, or if you say what are these ligands are the signaling molecules. They are the molecules released by the signaling cells that are responsible for transmitting information between cells and within a cell in the body. Now this means that these ligands or these signaling molecules, they are released in the body when they are required, and they are actually released by the signaling cells, which are used for communication between the cells. That means you're talking about the intercellular signaling, or these ligands can be responsible for the intracellular signaling, that is within a cell in the body. Now these ligands, they actually interact with proteins in the target cells, especially when you talk about the intercellular signaling. So these ligands, they will be released by these signaling cells. They are going to interact with the proteins in the target cells, and these proteins in the target cell to which these ligand interacts, they are actually called as the receptors. So generally these ligands and the receptors, they exist in several varieties, and these varieties of the ligands and the receptor, that is actually responsible for the communication in the living world. But a specific ligand is only going to interact with a specific receptor, and that is going to determine the specificity in communication between the cells or within a cell. So that means that a specific ligand is going to interact with a specific receptor. A ligand cannot interact with all of the receptors in the body, or a receptor can interact with all of the ligands available in the body. So this interaction between the ligand and the receptors, that will be specific, and that is going to generate a specific response in the cell. Now these ligands or these signals, they can be of different types, and you can actually classify them into different types based on different criteria. Now one of the criteria to classify these signals is based on their location in the body, that where these signals, they are present. So you can actually classify the signals based on the location. When you talk about the location of these signals, there are actually two basic types of the signals, and these signals are responsible for regulating the molecular and cellular activities, as well as cell-to-cell and cell-to-matrix communication. One of the types of the signal based on the location is called the extracellular signals. And as the name indicate, these kind of the signals, they are present in the extracellular space, and they serves as signals that initiate and control molecular and cellular activities. So this extracellular means that these signals are present outside the cell, they are not present inside the cells. There are different subtypes of these extracellular signals. One of the subtype are called the biochemical molecules, and actually the cell secreted factors and the extracellular matrix factors such as the ions and the proteins, for example the interferons and the steroid, they are actually included in these biochemical molecules which are the extracellular signals. Another type of these extracellular signals, they are called is the physical factors. Now, sometime in the body, there mechanical forces are there, and they are responsible for the deformation, or they are responsible for the mechanical sharing of the body of the cells. And these mechanical forces and deformation, they are actually responsible for different cellular activities, which range from the migration of the immune cells to the damaged area towards the apoptosis, when the cells they are not recoverable. The process of the apoptosis is actually going to remove the cells from the body and replace them by the new cells. So these physical factors, they are present outside the cell, so we put them under the extracellular signals. The third type of the extracellular signal, they are called is the electrical signals. And actually the neurotransmitters, they are responsible for the generation of action potential, and these action potential are generated by these ions which are present in the extracellular matrix, and these ions are going to generate the neurotransmission between different neurons. So these electrical signals they are carried out by these ions, and these ions are present in the extracellular matrix. They are present outside the cell, therefore we put them in the extracellular signals. So this is one type of the signals based on the location, the extracellular signals. The other type of the signals based on the location, they are called is the intracellular factors or the intracellular factors, and as the name indicate, these are the intracellular, that means they are present inside the cell. So these intracellular factors are present in the cytoplasm, and these intracellular factors actually serves as element for the signaling pathways. We'll be discussing different kind of the pathways, and these elements, they will be very much clear in these particular pathways, that these intracellular factors are actually the elements of the signaling pathways, thereby regulating different cellular activities. So the general rule is that an extracellular signal that must cooperate with intracellular signals to initiate and control cellular activity. Now, it doesn't mean that the intracellular factors or the extracellular factors, they are alone enough to carry out a particular signaling pathway and create a particular response in the cell. The extracellular signal, they must cooperate with the intracellular signals. So it is actually the combined activity of the extracellular and the intracellular signals which initiate and control cellular activity. These intracellular factors are also known as the regulatory factors, and they actually includes the proteins, for example the ion channels, the protein kinases and phosphatases, the lipids and the ions, etc. So as these proteins, these lipids and these ions are present inside the cell, therefore we call them as the intracellular factors. Another way you can classify the signal is based on the polarity, and based on the polarity you can actually classify the signals again into two types. One type of the signal, they are called is the hydrophobic ligands. As the name indicates, these kind of the ligands, they will be hydrophobic in nature. They are not going to like water, therefore we call them as the hydrophobic ligands. And if you talk about the important members of this class of the ligands, they are the steroid hormones. So these steroid hormones, they are actually examples of the hydrophobic ligands. These hydrophobic ligands, they have got specific properties. One of their property is that they are lipid in nature, and they have a hydrocarbon skeleton with 4 fused rings. As you can see over here, that they are having these 4 fused rings. These are just some of the examples of the hydrophobic signal that we will be seeing later on. So these they are lipids. They are having a hydrocarbon skeleton and with 4 fused rings. These hydrophobic ligands, they directly diffuse through the plasma membrane and interact with internal receptors. As most of the part of the plasma membrane that is hydrophobic in nature, that means that these hydrophobic ligand can easily diffuse through the plasma membrane. Because the plasma membrane is hydrophobic, the ligands, they are hydrophobic, they are going to like each other. So the plasma membrane is going to allow these hydrophobic ligand to diffuse directly through the plasma membrane and interact with the internal receptors. This term internal receptor, that will be clear in this video. We will be discussing that under the category of the types of the receptors. Another important property of the hydrophobic signals is that in order to be soluble in the blood, as the blood is hydrophilic media, so in order to be soluble in the blood, these ligands must bind to carrier proteins, and these carrier proteins are actually working as a transporters for these ligands to reach to their destinations. One of the important properties of the hydrophobic signal molecules is that they are generated by the cells, but they are generated on a demand from the plasma membrane lipids rather than stored in the vesicles. When you talk about the hydrophilic signals, the property of the hydrophilic signal is that they are produced by the cells, they are stored in the vesicles, and when they are needed, they are released in the body. When you talk about the hydrophobic signal, they are not formed earlier, they are not stored in the vesicles, but when they are needed, they are prepared at that particular time, and they are going to perform their function. These are just some of the examples of the hydrophobic signals. This is one of the hormone, the male hormone, testosterone. This is the cholesterol, and if you can see, they've got these 4 fuse rings. Now, this, if you look at this one, this is the plasma membrane of the cell, and the interior of the plasma membrane that is hydrophobic in nature. So these hormones, they are actually going to pass through these cell membrane directly, and they are going to interact with the intracellular receptor proteins, and by interacting with the intracellular receptor proteins, they are going to change the gene transcription, thereby generating a cellular response. So this is actually a very general sketch which shows how these hydrophilic signals, they work. So they can directly pass through these cell membrane, interact with these intracellular receptor proteins, thereby altering the gene transcription and creating a cellular response. So another type of the ligands or the signaling molecules based on polarity, they are the hydrophilic ligands, or they are actually the large molecules. Now, as they are hydrophilic in nature, that means that they cannot pass through a cell membrane, and in order to receive or in order to pass their message to the inside of the cell, they have to rely on the surface receptor proteins. So these ligands are going to interact with the surface receptor proteins, and once the receptors once the surface receptors, they get activated, they are going to generate a signal transduction pathway. If you talk about the ligands in this particular group, they are quite diverse, and they include small molecules, the peptides, and the proteins. Now, this is one of the important property of the hydrophilic molecules, that these hydrophilic signals, they are generated by the cells that release them, and they are stored in vesicle until they are required, or they are required for the cellular signaling in the cell. So this is in contrast to the hydrophobic signals. The hydrophobic signal, they are generated at that particular time when they are needed. The hydrophilic signal, they are produced, they are stored in the vesicles, and when they are needed, then they are released into the body. These are just some of the examples of the hydrophilic ligands. This one is the gamma aminobutyric acid, popularly known as the GABA, and this GABA is synthesized from this amino acid which is called is the glutamate, and this is the structure of the GABA. This is dopamine is actually a neurotransmitter, and this dopamine, the structure of dopamine is like this, and this dopamine is indirectly synthesized from the phenylalanine, or it can be directly synthesized from the tyrosine. Now this image is actually showing you a comparison between the hydrophobic and the hydrophilic signals. When you talk about the hydrophilic signals, they cannot pass the directly cell membrane, so they have to rely on these surface receptors. So the hydrophilic signal, they are going to interact with these receptors on the surface of the cell, and then they are going to convey their message to the interior of the cells. When you talk about the hydrophobic signals, they can actually interact, they can actually enter directly into the cells without the need of a cell surface receptor. This is a peptide hormone, the popular insulin, which is actually responsible for regulating the blood glucose in your body. And if you look at this one, this is comparatively a larger ligand. This GABA and this dopamine, they are smaller molecules, and this insulin, this is actually a very bigger kind of the molecules. This peptide hormone insulin is made up of two chains. One is called is the A chain, the other one is called is the B chain, and this A and the B chain, they are actually linked to each other by these cysteine bridges over here at this particular position, and at this particular position the cysteines of the chain A and the B, they are going to interact with each other, making the cysteine bridges. So these are the interchain cysteine bridges. There is also one intrachain cysteine bridge in the chain A, where the cysteine, one cysteine in the chain A is going to interact with another cysteine in the same chain A. Thereby, this particular cysteine bridge is known as the intrachain cysteine bridge, and these are known as the interchain cysteine bridges. So, another important component of the cellular signaling, they are known as the receptors. Now, regardless of the nature of the signal, whether they are the hydrophobic or they are hydrophilic in nature, these particular signals are going to interact with a specific protein, which is known as the receptor. And this is actually the interaction between the ligand and the receptors that is responsible for generation of a cellular response. Now, in most of the cases, not all of the cases, in most of the cases, the receptors, they are the transmembrane proteins. A transmembrane protein is a protein that goes from one side of a membrane through to the other side of the membrane on the target cell surface. So this means they have got domains outside the cells, and they have got domain inside the cell as well. So most of these receptors, they are the transmembrane proteins. And these receptors, which are transmembrane proteins, they have got three specific domains. The first one is called is the extracellular domain. The other one is called is the transmembrane domain, and the third one is known as the intracellular or the cytoplasmic domain. If you look at this figure, that is actually showing you the three domains of a transmembrane receptor. This, if you can see this one shown in the red, represented by this E, is actually known as the extracellular domain of the receptor. And the function of this extracellular domain is to interact with the ligand. This second domain of this receptor is known as the transmembrane domain, and the function of this transmembrane domain is to provide stability to this receptor, and this is actually present in the interior of the plasma membrane. And the third domain, which is known as the intracellular or the cytoplasmic domain, that is actually present in the cytoplasm over here, that is present in the interior of the cell. And the function of this intracellular domain is to make a bridge between the extracellular and the intracellular domain to actually transfer the message from the extracellular environment to the intracellular environment. If you talk about the types of the receptors, they are broadly divided into four types. One is known as the ion channel linked receptor, and the other one is the enzyme linked receptors. The third one is known as the G protein linked receptors, and the fourth one is known as the internal receptors. So let us talk about them in a little bit detail. When we talk about the ion channel linked receptors, so as the name indicate, these kind of the receptors, they will be linked to some kind of the ion channels. So these ion channel linked receptors, when they bind a ligand, they actually open, and through this opening the ions, the specific ions, they can actually pass through. If you talk about these ion linked channel receptors, in order to interact with the phospholipid fatty acid tail that form the center of the plasma membrane, as they are the transmembrane protein, so this part, which is known as the transmembrane domain over here, this transmembrane domain, that is actually made up of many amino acids which are hydrophobic in nature, and the amino acids that line the inside of the channel, they are hydrophilic and allow the passage for the water or the ions over here. So if you can see over here, this is actually the plasma membrane. This is the transmembrane domain over here on both sides of the receptor. So these sides of the membrane, they are actually made up of the hydrophobic amino acids. So they are actually interacting with these hydrophobic environment of the plasma membrane. Now this interior of the receptor is actually made up of the hydrophilic amino acid, and these hydrophilic amino acids are actually providing a hydrophilic environment for the ions or the water to pass through, because these ions, the ions are hydrophilic in nature. The interior of the receptor, this one over here, that is hydrophilic in nature, so they are actually going to they are going to allow the passage of the ions over here. Now when there is no ligand binded to these receptors, so you can actually see that they are closed, and when they are closed, they are not going to allow the transfer of the ions from the exterior environment into the interior environment. But when the ligand bind to these channels, there are conformational changes. You can clearly see the conformational changes over here, thereby creating a channel through which the these ions, they can actually pass, and when they can actually pass, they can actually perform their function. So we will continue this discussion in the next video.